So we tried to model this disease simply by doing what other systems and other labs have done for breast cancer, for lung cancer, for liver cancer. And we systematically took out tumors, brought them back to the lab, dissected them and dissociated them, and tried to develop cell lines to actually study the disease in vitro. I did this for nearly 6 months, going to the OR, grabbing tissue, dissecting it, dissociating it, and trying to develop cell lines. And for the first time, we were able to develop 2 cell lines that very much recapitulated the disease. And let me show you what I mean by recapitulating it. So these are the cells that grew in culture. They have these very unique fossiliferous features, very much like what chordomas have. So this is a specimen that we took from the operating room, and you can see these bubbly features are hallmarks or pathognomonic for chordomas. And we introduced these cell lines to the literature, gave them names. We even deposited them in the chordoma through the Chordoma Foundation. We received a small grant, and these were the first 2 cell lines that were becoming commercially available for people to study on chordoma. We asked then the next question, and that is the Brachyury protein critical, for the survival of these cells, and how important was it in regulating that biology? So we characterized the cell line, did a karyotype. We started injecting the cell line in animals and showed that we can recapitulate the growth of the tumor. And here you see the xenograft growing in the animal, and you see here a histological view of the tumor from the patient in this panel, and the Brachyury expression, as well as the tumor from the animal xenograft, as well as the Brachyury expression. So this was the first animal model that recapitulated the disease simply from the cell lines that we were able to harvest. We next asked the question of trying to turn off Brachyury. And so we used shRNA as well as CRISPR technology to silence the protein. And you can see here from this Western blot, we had the Brachyury protein with a control vector, and this is with Brachyury silenced. We can completely turn off the protein. And look at what happens dramatically when you do that. So here are the control cells, here's the control vector. Look what happens to the shape of the cells in culture. I mean, this experiment was a huge paradigm shift for me, because for the first time, we were able to arrest this disease in culture. This is a proliferation assay showing that when you turn off Brachyury, you almost completely shut the proliferation of these cells down. And then this is a flow cytometry assay showing that the vast majority of these cells, when you turn off Brachyury, shifted from viable to an apoptotic. Nearly 88% were in the apoptotic population. We did microarray studies showing that when you knock down Brachyury, you can see a decrease in the stem cell genes, the anti-apoptosis genes, and an increase in epithelial markers, suggestive of a change in the physiology of these cells. And then ultimately, we did the really proof of concept, and that is we were able to in vivo prevent tumor formation in animals. And here in this experiment, in this proof of concept experiment, we turned off Brachyury in these mice that were harboring chordoma, and we showed that we abolished tumor formation capacity. So in a span of 10 years, we were able to model the disease, identify a legitimate target, and demonstrate that by turning this off, we can now arrest this disease. And today, there are clinical trials that are trying to target Brachyury through immunotherapy. There are clinical trials trying to target Brachyury through molecular targeting. And I was very proud to be kind of a part of this work, but it taught me so much about how we need to care about our patients and take these challenges that we see, despite the most beautiful surgeries that we can do, and bring them back to the lab to really understand them better.
Hey everyone, Ryan Rad here from neurosurgerytraining.org. If you like that video, subscribe and donate to keep our content available for medical students across the world.